Hi, I'm Neil Cobb from the Biodiversity Outreach Network, and we're hosting this webinar, which is part 2 of a webinar that Mike Sharkey is giving. Mike started his career as a research scientist at the Canadian National Collection of Insects. After about 10 years, he moved to the University of Kentucky, where he went through the ranks and became a full professor. Presently, he is a professor emeritus and lives in Southern California. He has several hundred publications to his credit, mostly on the taxonomy of Braconidae and the phylogeny of Hymenoptera.
All right, thank you, Neil. Once again, I will be talking about the Ichneumonoidea, which is comprised of 2 species-rich cosmopolitan families, the Braconids and Ichneumonid. The last talk demonstrated that our current approaches to describing the species of Ichneumonoidea are problematic and must change if we are going to deal with their vast diversity. Today's talk will be about one way to overcome this taxonomic impediment. I will begin with a description of the ingredients of our recent publication, in which we described 403 new species of Costa Rican Braconids. I will then talk about tweaks we intend to make in our next publication. Then I will respond to recent criticisms and finish by my vision of taxonomy in years to come. The point I just made was that what's exceptional about this revision that I did a while ago was that there's no morphology in it. There's no key, diagnosis, or description. Instead, there's a consensus barcode that acts as the diagnosis. It is a simple method, it's fast, and it's efficient. Most of the criticisms to this approach will be addressed later in the talk, but there is one that I will mention now. The consensus barcode is included only to satisfy the code. It is simply an average of all of the barcodes that belong to the species. For example, if a species is polymorphic at a particular site, a UPAC code is placed there to indicate the polymorphism. The revision was meant to describe the species that were reared and Malays trapped in Costa Rica, and the barcodes that I generated often contain specimens on board that I did not view and were not from Costa Rica. In the future, we will simply use the consensus barcode based on those specimens that I have examined. As I mentioned, the consensus barcode is there only to satisfy the code, not to circumscribe the species. Creating a robust species concept from a handful of specimens in Costa Rica is not going to happen for most species. Much more collecting and barcoding from many areas is necessary to make this happen. Rather than using the consensus barcode, here's an example of how I perceive species identification to proceed with these preliminary species concepts. Let's see if I can get that going. Yes. Stantonia Henry Kekmani is a fair example. In this bin, we see 2 distinct clades on the neighbor joining tree. We can be reasonably sure that all of the specimens in the lower clade are part of the species, since the holotype is there. However, if one was to barcode a new specimen and it landed in the top clade, there would be somewhat less confidence. Situations like this are going to happen all the time until we get much more data, more specimens barcoded. This is a good time to mention that when we make a decision on a species limit, we carefully look at all specimens in the bin for morphological congruence and host data congruence, as well as comparing the species concept with specimens that are in neighboring bins. We don't simply rely on bins. Contrary to what is implied in the title of Meyer et al.'s paper criticizing our approach, we do not and did not simply equate bins with species. In fact, we were explicit stating that in our paper that bins do not and cannot be named. They are liable to change over time, splitting and disappearing altogether. And besides, they do not have a type specimen to peg the ownership of the bin. As they are presently formulated, bins cannot be named, and no one has tried to do this, ever. I mean, none of us have tried to do that, ever. At least to my knowledge. As mentioned in the previous slide, we looked closely at the specimen composition of each bin. In the case you see here, that was published in our paper, there are 11 species of Macrocentris in one bin. They all look different, and we distinguished 7 of them in a morphological key. We only described 7 species because what you see here is a newer tree that contains even more species in that same bin, and these were not available to us at the time. Basically, we see bins as a morphologically based taxonomist would view a tray into which they place their preliminary attempt at separating species. From this point, they would look more closely at morphology and host associations of the specimens, both in the tray and in neighboring trays, just as we did with specimens in the bin and neighboring bins. Of note here is that although the bin algorithm failed to differentiate these species of Macrocentris, the CO1 barcodes did not fail. Each species comes out in a distinct clade. When the barcode algorithm, I'm having trouble with my computer here, excuse me. When the barcode algorithm was first introduced, it was referred to as an approximation. No one would believe that an arbitrary 2% divergent cutoff is a universal biological trait. But it is amazingly good, in my opinion. And as demonstrated in the Sharkey et al. Paper, only a handful of the almost 400 bins had more than one species. Just as in phylogenetic analysis, congruence is a determining factor in using barcodes for species delimitation. In the case of this group of Macrocentris, morphology, host use, and sequence data are all congruent for each proposed species. This congruence is very, very unlikely to happen by chance. Barcode-based species delimitation is not a magic bullet. Like all other methods of species delimitation, the denser the specimen sampling is, the more likely one is to arrive at the correct answer. In this example, from our paper, we have one specimen from Western Canada and a handful of specimens from Costa Rica. The holotype, which is not barcoded, is from California. Morphologically, all specimens in the tree are similar. Although the specimens are in the same bin, there is a large gap between the Canadian and Costa Rican specimens. With this limited information, we cannot have complete confidence that these are the same species, and we have 2 choices. Here we took the more conservative approach, since both morphology and the bin algorithm suggest that they are the same species. Barcodes will not definitively solve problems like this without more data. For the next section of the talk, I will go over the 5 criticisms of standard taxonomic approaches that I detailed in the last webinar. These are that the rate of description is too slow, it's too expensive, the quality of species delimitation is poor with too much lumping and splitting, the keys do not work for megadiverse groups and they never will, and there is an overdependence on type specimens. In the previous talk, I gave detailed evidence that the number of species of Ichneumonoidea is well over 1 million and showed that we have described only about 5% of these. Moreover, since 50% of the described species are from the Palearctic, I showed that neotropical countries have less than 1% of their Ichneumonoid species described, just 1%. Based on our productivity over the last 20 years, I showed in the last talk that it would take more than 2,000 years to describe all the species of Ichneumonoidea. With a barcoding approach, it will be easy for an individual scientist to describe 1,000 species per year. And at that rate, it would take only 10 dedicated taxonomic positions 100 years to describe 1 million species. Again, in the last talk, I mentioned the effects that global warming is having on the ecosystems of Costa Rica. And as all members of this audience who are taxonomists know, there is urgency to catalog as many species as possible now, so that we have a baseline to document extinction for conservation purposes. The second problem that I mentioned in the last webinar is that our present approaches are too expensive. And of course, this is mostly because they are too time-consuming and labor-intensive. I show that it would cost about $63 million simply to describe all Costa Rican barconids using current approaches. Since the barcode approach is a magnitude more efficient, dealing with 1,000 species per year per taxonomist instead of 100 per year per taxonomist, the cost would also decrease by a magnitude. Thus, the Costa Rican barconids could all be treated for $6.3 million. That is a number that funding agencies can work with. By the same logic, all 1 million world species of Ichneumonoidea could be treated for $160 million instead of the astronomical $1.6 billion traditional approaches would cost. Since we are on the topic of cost, it is worth mentioning an article by Srivatsan et al. Entitled Min-ion Barcodes, Biodiversity Discovery and Identification by Everyone for Everyone. Here they describe the latest technology and workflow for obtaining CO1 barcodes for less than 10 cents each. As the title suggests, this will allow any lab anywhere in the world to afford barcoding. I have often heard that barcoding is unfair to those doing research in poorer countries, and it appears that this bottleneck is history. The third problem that I mentioned, one that is restricted to morphological approaches, is that species delimitations are very poor, with a 33% published synonymy rate and an even higher rate of lumping. In this slide from the last talk, I showed a real example of lumping, an error I made in a publication on the genus of barconids from Costa Rica. These images show 4 of a group of 5 cryptic species of Alabagras, and I lumped all these under the name Alabagras pachamama. Sequence data, supported by host data, discovered these 5 species. Once they were delimited by these data, subtle but consistent morphological traits were found to distinguish them. For example, the position of the dark bands in the forewings distinguished one species. Note that the band is below the stigma in the wing on the left, and it's on top of the stigma in the species on the right. Excuse me, I'm continuing to have trouble here. Yeah, it's just not working. There it is. The color of the legs distinguished the other species, some of the other species. These morphological traits were exactly congruent with the groups inferred by the CO1 barcode. In summary, the error rate when using CO1 sequence data is far lower than that of morphological data. I don't know what's wrong with my, there we are. The next criticism that I mentioned in my last talk was that keys do not work for hyperdiverse genera, like many of those in the Ichneumonoidea. What you see here is one couplet in a key that would have to be over 3,000 couplets long to deal with the estimated 3,000 species of Neotropical Mesochorus. Obviously, an impossible task. To identify a specimen with our method, one obtains the CO1 barcode. Remember, that can be now done for less than 10 cents. The FASTA file is entered on bold and a search is made. If there is a match, the match has a species name, and if the match has a species name, it will be given. That's point number 1 on the slide, that blew up a little bit. Also generated are all the exact matches and a list of the near matches in descending order. That's point 2 in the slide. This set of matches is also presented in graphic form, point 3 in the slide. Here's a readable look at the exact and close matches, or some of them. And each of these can be examined in more detail on bold by selecting the blue arrow to the right. If one goes to the bin page by selecting the blue box at point 4 in this image, All of the public records in the bin are available. A map of the distribution of the members of the bin is presented, as well as links to all of the specimen pages for members of the bin. In this case, Therophilus Penteado-Diese is found only in Costa Rica. Each of these records can, in turn, be investigated individually. If the first specimen in the bin is selected, the following page appears. Here you get an image of the specimen, a FASTA file, a distribution map, and locality information, as well as other details that I have not included. Going back to the first page that appears after one submits a sequence, there is also the option to select the tree-based identification. This is point 5 on the slide. Selecting this generates the following tree. The tree that is generated is much larger than this, and I have clipped a segment of it. On the lower right-hand corner in red is the submitted specimen. It is identical to 2 other specimens of Therophilus Penteado-Diese, so it fits the species concept. This species concept is the product of the first reviser in this case, but specimens are continually being added to BOLD. Taxonomists such as myself, and even technicians at BOLD will place names on them based on their position in the tree and on their images. So the species concept, such as it is presented on BOLD, is a dynamic, evolving, community-driven theory. In the case of Therophilus Penteado-Diese, all specimens differ by only one base pair. However, it is easy to imagine that the members of a species are more diverse. Especially in these cases, it is good practice to compare the identified specimen with the barcode of the holotype, which is here circled in red. And in this case, the submitted specimen and the holotype have identical barcodes. So we're very sure of the identification. The last criticism that I made with respect to morphological approaches is that for an accurate identification, it is almost always necessary to view type specimens at great cost in time and money, and a special handicap for those from many countries whose postal systems are not secure or whose customs practices are draconian. As previously demonstrated, if a holotype is barcoded, one can identify conspecific specimens with reasonable certainty and without borrowing the type or traveling to a far-off museum. Okay, I've now gone through the 5 criticisms of traditional approaches that I mentioned in my last talk, and how a barcode-based system improves on these. The paper in which we described 403 new species was the first large-scale attempt at using barcodes, and there will be modifications in our next efforts. One aspect that we will change has to do with nearest neighbors. On BOLD, the bin that is most similar to the bin containing a species that one is describing can be found on the bin page. Previously, we checked this bin for morphological and host use differences from the species being published, but we did not publish these findings. If you ask for the neighbor joining tree by using the link indicated by the red arrow in the bin page, you are presented with a tree that allows you to identify the nearest neighbor. The tree will look something like this, and the nearest neighbor in this case is identified as Orgylus scott-morii. We can see that there is about a 7% divergence between the 2 species, the one that we're submitting and the scott-morii. And that gives us considerable confidence that they are distinct species. You can then check the images of specimens on BOLD, or the actual specimens that you are treating. And here we see that Orgylus everett linguisti is, and its nearest neighbor, scott-morii, are quite different. For example, the species on the left is much smaller and differs greatly in color. In the last paper, we went through these steps, but what will change in the next paper and subsequent publications. Sorry, I'm having trouble here. Excuse me. It's just the computer's not responding. There it is. Well, basically, what we're going to do the next time is we're actually going to do a diagnosis, separating a morphological diagnosis, separating new species from their newest neighbors. So this will be a morphological aspect that we're adding to our CO1-based barcode approach. And this sage advice comes from my dipterist colleague, Brian Brown. As some in the audience may be aware, there was a recent preprint of a paper by Ahrens and co-authors entitled, "It is Time to Describe New Species Without Diagnoses," a comment on Sharkey et al. 2021. In this paper, there was a suggestion that our last publications did not fit the code, which I consider balderdash, and a not so veiled threat that barcoded based descriptions may not fit future changes in the code. This modification of a morphological diagnosis to distinguish nearest neighbors, besides being good practice, will probably put that threat to bed. One criticism that we hear often is that barcode-based treatments will create dual systems of taxonomy, one based on morphology and the other based on sequence data. This would be true if there was a morphological system in place. But we are not suggesting, for example, that researchers begin a barcode-based description of Palearctic crab beetles. We agree that they should be barcoded, but since the fauna is well known, new species should also be placed in existing keys and so on. We are suggesting that the barcode approach is for taxa that did not have a morphological system in place. As demonstrated in my last webinar, less than 1% of the neotropical Ichneumonoid fauna is described. There will only be one system for groups like this if we want to get it done before the year 4000. Furthermore, for the 1% of described species, the holotypes will eventually be barcoded, or topotypic conspecifics will be barcoded. So all will fit into the same system. Over the next few slides, I'll respond to some of the criticisms made in the recent preprint by Meyer and co-authors. First off, I will reiterate that we have always recognized that bins are not perfect and have never suggested that they be named. They do not have holotypes and are somewhat dynamic, capable of merging and splitting. So the title of Meyer et al. Is somewhat misleading, in my opinion. In the 2021 paper, we made the statement, we view barcode-based descriptions as a first pass in an iterative approach to solve the taxonomic impediment of megadiverse and under-taxonomically resourced groups. This is meant to suggest that most of the species concepts that we present are probably correct, and that if a future reviser wishes, for example, to write a key to the species of Zelomorpha of Costa Rica, they have a head start with the species well defined. It was also meant to suggest that it will not be perfect, and that more collecting and more barcodes will refine the species concepts. Meyer et al.'s suggestion that we should have collected and analyzed and published more data seems naive. The entire point of the project, of the approach that we have, was to speed up the description process. If more data means more collections, more sequence data, and the addition of more morphological descriptions, then the entire purpose is defeated. And we are looking at a completion date of the year 4000 again. Another point to remember, as demonstrated in my last talk, is that the keys to these megadiverse groups are never used. And when they are, they're usually resulting in misidentifications. Meyer et al. Made the claim that bins are unstable. This is not news to us and is why we do not simply name bins. To demonstrate instability, they compared the bins supported by the bold algorithm with three other methods. that have been devised to group species. The authors of these methods are highlighted in yellow above. They then showed that as you increased the size of the data sets, fewer bins agreed with the other tree methods of species delimitation. I simply find this argument without merit. If they use 20 different methods of delimiting species, the overlap would undoubtedly be close to zero. The four are different methods and are designed to be different. A valid comparison in phylogenetic analysis would be comparing a molecular marker like 28S with 3 other genes and suggesting that 28S is a poor phylogenetic marker because it was not perfectly congruent with the other 3 markers. As you added markers in taxa, you would continuously find less congruence. What their comparisons mean is not understandable to me or to anyone I talk to. Meyer et al. Noted that there are 11 cases in the 2021 publication where the barcodes coded for identical amino acids and came to the conclusion that barcode clusters should only be treated as a first pass, but not described. Despite the fact that we also use morphology to differentiate species, we thought it best to investigate this criticism since we paid little attention to amino acids previously. Austin Baker, a recently graduated PhD student from John Harady's lab, investigated several cases in which different species have barcodes that code for identical amino acids. He started with the genus Litopilus, where he found a well-sampled example where 3 bins with identical amino acids were treated as 3 different species. You can see here the beginning of the CO1 alignment showing the codons colored for the amino acids they represent. To the left are the 3 bins, which we recognized as species. The top one is blue, the middle one is pink, and the bottom one is yellow. When we color the matrix by nucleotides, we can easily spot the so-called silent mutations in 3rd codon positions that are autapomorphic or diagnostic for each of these 3 species. These positions are marked with arrows, and the arrows are colored for the taxon that is unique for that position. For example, the first yellow arrow on the bottom left points to a 3rd position base pair autapomorphy for Litopilus octune, which is the species on the bottom left. The other arrows point to autapomorphies for the blue and pink species. By the end, we found 4 autapomorphies for the blue species, 2 for the pink species, and 5 autapomorphies for the yellow species. There is a repeated pattern separating these species across positions with silent mutations. And there are a few places where we find polymorphic 3rd positions within a single species, such as the one indicated by the long white arrow here. In fact, there are only 4 such positions among all 3 species. So let's check the additional evidence and see and that we use to distinguish the species, morphology and host data. All 3 species are on different host caterpillars. And any morphologist would consider these distinct species based on a suite of characters. As you can see from the map in the right-hand corner, they were collected from multiple locations, and they showed the same pattern of CO1 and morphological difference across localities. Let's have a closer look at the morphological differences. The wing color patterns are different in all 3 species. That one in the middle of this slide is particularly distinct. The body color of the species on the bottom is completely different from that of the other 2 species, and the sculpture of the podium is distinct in all species. Dr. Baker is going through all of the published barcoded specimen species to check for potential synonyms when amino acids are identical, but he has not reported any of these to me yet. Depending on the results that Dr. Baker obtains, we may start to check species pairs with identical amino acids, but there's no evidence yet to do that. Building on the same theme of bin instability, Meyer and co-authors compared the number of bins that are on bold presently to the number of species presented in various publications. On the right-hand column here, you see the deviation of the two, the number of published species and the number of bins that are there today. I did not have time to go through all of these, but I am somewhat familiar with the revision of Apantales by Jose Fernandez. In the Apantales publication, Dr. Fernandez did not use a barcoding approach for his species concepts. His species concepts in that publication do not match bins any more than they do now. There is no mention in his species descriptions of bins. In fact, knowing something of his work, he relied much more heavily on morphological data and to a lesser extent on rearing data. I'm not sure if bins for species of Apantales are not very informative or if Jose should have paid more attention to the bins. It would take weeks for me to figure this out. In any case, the comparison of species to bins in this example is totally misleading, since the difference was probably 25% or even more when Jose and co-authors published in 2014. The only way that I could reliably verify or refute Meyer et al.'s claim of bin instability over time was to take a closer look at our 2018 revision of Costa Rican Alabagras. I know that we relied heavily on bins and morphology in the same way that we did in the 2021 paper that he's criticizing. There has been 4 years since the species concepts in this Alabagras paper were finalized, and Dan and Winnie and their team have been adding specimens to Bowl continuously since then. Of the 80-odd species in the tree that I generated a few days ago from Bowl, not one species concept changed. There is one species, Alabagras Sharkey, that occupies 3 bins. This is exactly as it was in the original publication, and it was morphology and host use data that convinced us that the binning algorithm was incorrect in this one case. In conclusion, the bins have been completely stable over the last 4 years. Over time, with even more sequence data, there may be some Alabagras species concepts that change. This is what you expect with more information. And that is one of the reasons why I do not promote the idea of exhaustive keys and morphological descriptions for understudied hyperdiverse groups. It will take years to come to a point of complete stasis, if ever. Here in the United States, and I expect in most other countries, it is all but impossible to get major funding for descriptive taxonomy. Barcoding projects, however, are receiving major funding at the level of tens of millions of dollars. Besides the BOLD initiative in Guelph, there are many other large, well-funded initiatives. Here are 3 examples. Barcode 500K, an initiative by the International Barcode of Life, the BioAlpha project in Costa Rica, and Life Plan, a worldwide initiative. Barcoding has been used as an educational tool in many high schools and universities. In the past, students have been using Sanger sequencing and then searching on BOLD for identifications. Now it is possible to do real-time sequencing in the field with the new Oxford nanopore technologies. And these technologies will only get better and cheaper. Initiatives like the Finnish barcoding project will allow for the majority of their arthropod fauna to be identified by anyone. No keys, no descriptions, no expert taxonomist involvement. I want you to imagine a future in which school children can go in the field and identify arthropods in real time and search online to learn all about it. This sort of educational use will bond children to their environment in ways that may be transformative for future conservation efforts. A valued colleague of mine stated that she did not want to see barcode-based taxonomy because she does not have the capability to produce barcodes. The point is that without barcodes, the neotropical fauna will remain less than 1% identifiable for the foreseeable future, certainly for the rest of her life. Governmental identification services are already using barcode-based technologies for invasive species and such. In the future, most all identifications will be handled with barcodes, and most will be done by the scientist requiring the identification rather than by a service. When the database is complete enough, it will be easy to determine if a species is new to science. And if the person asking for the identification needs a name on it, it will only take a few hours to put together a published description, such as the one you see here. This talk is focused on the Ichneumonoidea, but they are not alone. Many other groups of Hymenoptera are hyperdiverse. The red accumulation curve shows the number of bins, a close approximation to the number of species, that have been captured in a single stationary Malaise trap in Costa Rica over 2 years. Note that this is not a theoretical species accumulation curve estimating the number of species. These are real specimens, real species that have been captured and barcoded. More than 3,000 species of Hymenoptera have been captured with no indication that the diversity is leveling off. The Hymenoptera are not alone. The same trap collected more than 6,000 species of Diptera. Yes, fellow Hymenopterus, we thought for a while that we had taken first place from Coleoptera, but Diptera are far more diverse than both of those. More than 6,000 species in one place. No approaches for taxonomy that are in place today can deal with this sort of diversity. As we humans inch into the future, technology is rocketing and at an accelerating pace. The Barcode of Life database in Guelph, Ontario is the center of this technological revolution in taxonomy, thanks to the enormous efforts of Paul Herbert and his team. Gold is an essential resource, and it is evolving dynamically. I expect changes in the near future that will allow us to proceed with CO1-based descriptions at a much faster pace than at present. This could be done with a workflow incorporated into Bold that would allow for semi-automatic descriptions. There would still be work for taxonomists to proof the results and place the specimens at the generic level. But the tedious process of copying locality data, creating consensus barcodes, maps, identifying nearest neighbors, and such would eliminate 90% of the effort. Since time is the largest expense in the way of fixed costs like salary and overhead, speeding up taxonomic descriptions with such a workflow will save millions of dollars. For example, we could see the cost of describing the world species of Ichneumonoidea decrease by another magnitude, going from $160 million to $16 million. Advances like this are the only way we're going to describe a large portion of the 30 or more million undescribed arthropods. With that, I'd like to thank Austin Baker, Brian Brown, Dan Jansen, and Neil Cobb for their help. And for those of you that are interested in the literature related to barcode-based descriptions, here is a list, including a link to my last presentation. Note that the last two citations are preprints and might change. The talk will be posted as well so that you will have the opportunity to look at these citations in greater detail. And with that, I'm through. Thank you.
Thank you, Mike. And as I said at the introduction, if you could type your question, that would be great, because then we'll have a record of it. And I'll just read off the questions as they come in. Is there a question? Okay, in some taxa, there are hundreds of superficially described species with no barcodes and no specialist who has had the opportunity to examine the types. How would your approach handle the abundance of historical names in such groups? And that's from Jessica Awad.
Oh, that's a great question. I've given a lot of thought to that. There's really only one neotropical group that has had hundreds of species described, and there are tens of thousands of species in this group. And it's one group that I'm going to ignore for a long time in the hopes that museumologists will barcode all of those types before I get started on actually doing barcoding with fresh specimens and publishing new species. If they were to never barcode those holotypes, my belief is that eventually those types will be rather useless. And I would go ahead and describe all of those species, all of the fresh species. And if they ever decide to barcode the holotypes, then there might be a few synonyms. If over hundreds of years they never barcode the holotypes, I suspect some of those holotype names might become nomen nudums. But we'll have to see how the whole process evolves over the next decades to be sure of what's going to happen. The bottom line to the question is, what would I do with those groups? Well, I would ignore them for now.
Okay, Jonathan Coddington asks, he missed the first presentation. What exactly about the code requires a consensus barcode?
Well, at some point you have to distinguish the species you're describing from some other species. A diagnosis is essential, and the consensus barcode is a way to distinguish the new species from all other species. I'm not sure. Did I answer the question? Or?
Jonathan just says, so it's the diagnosis.
It is the diagnosis.
Yeah. Mike, but obviously the consensus barcode changes over time. I mean, I can compute the consensus barcode now and I can compute it in 10 years. So it is just sort of a trick to get the name into the literature.
Oh, absolutely. Yeah, I think it has almost no value at all. I was trying to make that point early in my talk. You know,
yeah, I just wondered what the, you know, the.
Yeah, it has no value at all. It's just a way to circumvent the code.
Okay, Derek, can you just ask your question, because it's not really clear from what you typed in there exactly what you're asking.
I wasn't asking. I was just clarifying that a consensus barcode is basically an attempt at including all the variation that represents the species, right? So when you're describing something morphologically, you have a choice. Do you want to diagnose like just based on the holotype? If you have very little variation, you're just focusing on the holotype, or are you going to try to encompass, you know, the variation in the species? The consensus is an attempt to encompass that variation that is known. And as Brian points out, as you know, if you're doing morphology-based work, as you get more specimens, the variation increases and the diagnosis will need to change. So there's no real, I don't think there's any real difference between a consensus barcode and the traditional approach.
Yeah, I agree with you absolutely in that regard. Nonetheless, there was the criticism that we were using a consensus barcode with specimens we hadn't even looked at, and that were in disparate localities. So in the future, we're going to restrict ourselves to a consensus barcode just for the specimens that we've observed and we're confident are the same species.
Let me add something here. Way back in 2003 and 2004, when Paul put this whole thing out on the table and the blackboard, that question immediately arose. And what we found was that the grouping in an NJ tree that you can visually see covers a lot of that. In other words, the thing you see on the screen of, say, 23 specimens on the NJ tree will show a small amount of variation. When you move that up to 100 specimens, you get a little more. That does not change the clump in the NJ tree. But if one of those specimens, or 2 or 3 or 5, turn out to have very different food plants, or very different host caterpillars, or very different behaviors, or very different places to live, what you want to do is emphasize getting more of those, and all of a sudden you discover that makes a distinct lump in the NJ tree. That then, if you take and pool those barcodes, you get a consensus barcode for that lump. You can also pool the entire lump, but if you pool the 2 different sets of barcodes with their 2 different biologies, and you put those in front of a really good hardcore morphological taxonomist, almost invariably, he or she finds nice morphological differences that you can tell those 2 apart. I could give you thousands of examples like that from here, from the ACG. If you try to add Peru or Mexico into that same pool, of course you get more variation. And for that, we need a lot more specimens from a lot more places. But we're not going to get them the old way we've been doing them. So what we're feeling is that if we can get the attitude in taxonomy and in biodiversity documentation to be collecting and storing and barcoding large numbers of individuals, pretty soon we'll be able to tell you whether the one in Peru that only differs by 1% of its barcodes is in fact different from the one in Costa Rica. Because all of a sudden, when you hit southern Panama, it flips from one to the other. And if you do your biology in the field, and we've been doing here in the ACG, you discover that not only do they bump into each other on a geographic map, but you can actually catch the 2 side by side, and they tell each other apart, and you don't get mixed barcodes except very rarely. We do have a number of those joining situations here in the ACG, which we simply documented by having large numbers of specimens. And each time we publish them, they are studiously ignored. So I'll just leave it there. But what what Mike and Brian and others are digging into is the way we can do the same thing we've been doing morphologically for 300 years, is ignoring what we think is intra-specific variation when we write a description or apply a name or work with a particular insect. And what we just find is that when we do the barcoding of those same things, we find this is really just one more very good character. And from my standpoint, it's a morphological character. If you had eyes that could see the DNA string inside that thing long ago, that would have been included as part of a standard morphological description, and its variation would also have been a bugaboo for you, just like morphological variation is a bugaboo. Okay, so I'll stop there. Thank you.
Okay, thanks, Dan. John, you want to just go ahead and ask your question, even though you typed it?
Yeah, just like you had that nice little tree where you put that nice little red circle around the holotype. And I wonder if there's any effort on the part of BOLD that when you pull up those bins that you can actually tell when something is the sequence of what has been represented as a holotype.
That's a really good question, John. We've actually, Dan and I both have asked BOLD to incorporate that into one field or another. And we're still working on the best field to enter that the type species. What was suggested to us was to put the holotype information in with, I think it's called other info, a field called other info. And unfortunately, Dan's host data is in the other info field. So it would read holotype and then the name of the caterpillar instead of the holotype. It would confuse people. So what I have suggested the last time Dan and I talked, my suggestion was to have the holotype actually on the name of the species. So the species name would have holotype incorporated into it so that you couldn't help but notice which specimen is the holotype and wouldn't have to ask for a special field to see that. But it's essential in my mind.
And also, I mean, even if holotype, paratype, I mean, if you're talking about accumulating these things over time, it'd be nice to know what's added as new versus what was added as part of the actual, description as you're putting together.
Yeah, that's a good idea.
Okay, are there any other questions? I don't see any questions written.
I could just ask one quick one because I've heard this said. I don't really advocate it, but I've heard people say, why give names at all? Why not just refer to them as bins, right? I know you've already argued that bins don't equal species, but they're fairly close. And some people who have a problem with this turbo taxonomy approach have asked that question. So I'm curious what your answer would be.
Well, there are two problems. The whole system of taxonomy is meant for information storage and retrieval. And if you don't have a fixed name on a species concept that people can write about and talk about, then you can't communicate information and you can't store information well. The bins do not have a holotype. So when you talk about this bin representing a species concept, when that bin changes and some specimens are split off of it and some specimens are added, or if it merges with another bin that had a name and then you lose the name of the bin, it's just massively confusing. And as such, it is not appropriate to store information with.
Thanks.
May I add a word to that? We tend to think as biodiversity scientists and as taxonomists, which I'm not one, that these terminologies are generated, built, used, developed within our own silo, within our own guild. But in fact, if all we think about is our own silo, our own guild, in a few years, a few decades, there won't be any of this material for us to work with. Whatever terminology we adopt has got to work with society as a whole. And just as you don't go around calling yourself by your social security number, you call by a name, and that name relates to the rest of your relationship with society. So if we start adding just numbers, just cold codes, you and I can take care of that. We can handle that. But other countries, other languages, other societies all have a species concept more or less in their minds. And somehow we have to adjust our terminology so that it relates to that adjustment in their minds. Other languages, other societies, other viewpoints as to what's a species. And so somehow we have to take that into consideration at the same time we build our own system for communicating amongst ourselves and amongst our own guild. So I'll stop there. Thank you.
Okay, are there any other questions? We're 5 minutes past the hour. But if there are any other questions, I'm sure Mike can answer them. Okay. If not, thanks a lot, Mike, for giving this two-part webinar. I appreciate it greatly. If you have any questions, feel free to either email Mike or myself, and we will post this webinar on the a number of places, but including the iDigBio website that Mike had in his last slide. And with that, I will